At the bottom of the Mariana Trench, the pressure is roughly 1,000 times what you feel standing at sea level. A human body would not survive a second. And yet, something lives there, not merely survives, but grows, reproduces, and actually requires that crushing weight to function. Remove the pressure, and the organism dies. That is not a metaphor. It is a documented biological reality, and it forces a question that cuts to the heart of how we define extreme. Extreme for whom? The deep sea begins, by scientific convention, at 1,000 meters below the surface. Below that threshold, sunlight is gone. Temperature in most of the deep ocean holds between 1 and 3 degrees Celsius, just above freezing, sustained across the largest continuous habitat on Earth. And pressure climbs at roughly 1 additional atmosphere for every 10 meters of depth. By the time you reach the seafloor of a Hadal trench, the deepest oceanic zones, below 6,000 meters, you are looking at pressures exceeding 600 atmospheres. At the very bottom of the Mariana Trench, pressures approach 1,100 atmospheres, or roughly 110 megapascals. That number is not an abstraction. It is the force per unit area pressing on every cell membrane, every strand of DNA, every enzyme in every organism living there. For most life, pressure at that scale is destructive. It compresses cell membranes, reducing their fluidity and restricting their permeability. It shifts the physical equilibrium of molecules towards states that occupy smaller volumes, which distorts the shapes of proteins and disrupts the precise three-dimensional folding that makes enzymes functional. It raises the temperature at which DNA transitions from its stable double-stranded form to the single-stranded configuration required for replication and transcription, making the basic act of copying genetic information more difficult. High pressure does not just challenge life. At the cellular level, it systematically attacks the machinery life depends on. And yet, a category of organism not only tolerates this, it depends on it. Scientists call them piezophiles, from the Greek for pressure and for loving. The formal definition is precise. An organism whose maximum rate of growth occurs at a hydrostatic pressure of 10 megapascals, or above, when tested across all permissible temperatures. The original scientific term was barophile, from the Greek for weight, but the field shifted to piezophile because the prefix more accurately describes what is actually being measured: hydrostatic pressure, not gravitational load. That distinction matters because the mechanisms these organisms have evolved are not responses to weight. They are responses to the physical chemistry of compression. Within the broader category, a subset pushes even further. Hyperpiezophiles are defined as organisms whose maximum growth rate occurs above 50 megapascals, roughly 500 atmospheres. And the current documented record belongs to a bacterium called Colwellia marinimaniae, strain MT-41, which was observed growing at 140 megapascals. That is approximately 1,400 atmospheres of pressure. No other organism has been confirmed growing under greater hydrostatic pressure than that. The first confirmed obligate piezophile, an organism that cannot grow at normal atmospheric pressure at all, was a cold-adapted bacterium, also from the genus Colwellia, designated strain MT-41. It was isolated from a decaying amphipod of the species Hirundelia gigas, recovered from the floor of the Mariana Trench. The organism's optimal growth pressure is 70 megapascals at 2 degrees Celsius. Below 20 megapascals, it cannot grow. That is the line that defines obligate. Not merely a preference for pressure, but a hard biological requirement for it. Bring this organism to the surface, reduce the pressure to what we call normal, and you have not rescued it. You have killed it. A separate discovery extended the picture in a different direction entirely. The first thermophilic piezophilic archaea, an organism that thrives at both high pressure and high temperature, was Pyrococcus yayanosii, strain CH1, isolated from the Ashadze hydrothermal vent site. Its optimal growth conditions sit at 52 megapascals and 98 degrees Celsius. Like its cold-adapted counterpart, it cannot grow at pressures at or below 20 megapascals. Two organisms, separated by nearly 100 degrees of thermal preference, sharing the same fundamental pressure dependency. That convergence tells scientists something important. The adaptations required for life under crushing pressure are not locked to any single environmental niche. They have emerged independently across thermally opposite ecosystems. How do these organisms actually manage it? Piezophiles regulate gene expression in response to pressure changes, effectively switching on different biological programs depending on the hydrostatic load they detect. They modify their cell membranes to maintain necessary fluidity under compression, a problem because increased pressure drives membrane lipids into tighter, less permeable arrangements. The adaptations required to counteract this are structural, written into the organism's biochemistry. Their biomolecules, the proteins, lipids, and nucleic acids that carry out every cellular function, are tuned to operate under conditions that would denature or disable their equivalents in surface organisms. The piezosphere, the volume of Earth's biosphere subject to high hydrostatic pressure, is enormous. It includes the entire deep sea below 1,000 meters and extends into the deep subsurface, which can reach 5,000 meters below the seafloor or continental surface. In terms of sheer volume, the majority of Earth's biosphere falls within this zone. The deep sea is dominated by cold-adapted pressure organisms, Hyperpiezophiles. Hydrothermal vents and the deep subsurface host Thermophilic, operating at temperatures above 45 degrees Celsius. Scientists have documented both heterotrophic metabolism, organisms consuming organic matter, and autotrophic fixation, organisms building their own organic compounds from inorganic sources, within this pressure-dominated biosphere. The organic matter available at depth is largely composed of refractory complex polymers, materials that have resisted degradation as they sank from the productive surface ocean. Metabolic research also points towards significant activity involving iron-bearing minerals and carbon monoxide. But scientists are direct about the limits here. The study of how piezophiles acquire nutrients and sustain metabolism is, in their words, still in its infancy. That phrase deserves to sit with you for a moment. The largest habitable volume on Earth by measurement, and the basic question of how the organisms living in it feed themselves remains incompletely answered. Not because the science is careless, but because the environment is genuinely difficult to access and nearly impossible to replicate. Samples retrieved from Hadal depths experience depressurization the moment they begin rising through the water column. Instruments must be designed to measure and preserve conditions that are hostile to the instruments themselves. Culture experiments have to maintain pressures that no laboratory bench equipment was built to sustain. Every verified data point about piezophile biology represents an engineering achievement alongside a scientific one. The unresolved question at the center of this field is not whether life exists under extreme pressure. The evidence for that is clear. The open question is mechanistic. Precisely how do obligate piezophiles sense pressure and coordinate a whole organism response to it? Gene expression changes with pressure, that much is established. But the molecular sensors that detect the change, the signaling pathways that carry that information to gene regulatory machinery, and the full suite of proteins that shift in response, these remain only partially mapped. The system exists, Its detailed architecture is not yet fully described. Pressure is the defining condition of the majority of Earth's living space. The organisms built to require it are not edge cases or curiosities. They are inhabitants of the dominant habitat on this planet, adapted over evolutionary time to conditions that would disassemble any cell brought up from the surface. The deepest confirmed growth, 140 megapascals, was recorded not in a thought experiment, but in a laboratory culture derived from a real organism recovered from a real place. The number is verified. What produced it, at the level of molecular mechanism, is still being worked out. And if the largest biosphere on Earth is still yielding its first obligate extremophiles, still mapping its metabolic pathways, still searching for the sensors that make pressure a signal rather than a threat, then the inventory of life itself remains genuinely incomplete. Not in the realm of speculation, in the realm of documented, measurable, ongoing discovery,